Hi, welcome to our lecture on mitosis. This is part 2 of our lecture on chapter 15, which includes the cell cycle, mitosis, and meiosis. Mitosis is division of the nucleus in preparation for full cellular division. When our cells need to divide for repair of damaged cells, or we could use division for replacing cells that may be lost, and also for organismal growth, the cell has to go through mitosis and cytokinesis. So mitosis is division of the nucleus, and cytokinesis is division of the cytoplasm and cell membrane. During this cellular division, during mitosis, we need to ensure that the sorting process of our chromosomes, that we get the correct number of chromosomes and combination of chromosomes in each of what are known as daughter cells. So during cell division, we start out with a parent cell. So we have a single parent cell, and then that parent cell will divide and become two daughter cells. So these are the daughter cells, and this is our parent cell. In order for this sorting process to occur correctly, we're going to take a look at some structures known as the mitotic spindle apparatus. It's usually just called the mitotic spindle. And this particular apparatus is composed primarily of microtubules and actin filaments, and it's used for that actual organization of chromosomes as well as the actual sorting or division process itself. So in animal cells, we have three different types of microtubules that create our mitotic spindle. We first have what are called astral microtubules. The astral microtubules you can see right here in this picture are up here, and they're at each end of the cell. And those astral microtubules come from the centrosome. So remember, centrosome, we actually, right here, right? So this is the centrosome. We have one centrosome that's normally in the center of our cell. Just prior to mitotic division in the cytoplasm of the cell, we're going to have replication of the centrosome. And we have one of each copy of the centrosome goes to each end of the cell. So it kind of starts to pull the cell into two different sides. These astral microtubules, are what anchor the centrosome into its poles. So it actually is what initially creates the two different poles. You could say that this is south and this is north. That's not really what they call them, but we have two opposite poles of the cell because we're going to pull this cell apart and create two out of it. Next to that, after astral microtubules, we have polar microtubules. Polar microtubules are what actually create the two sides of the cells. So these also come from our centrosome, and you can see here are our polar ones. Let me change my pen color. That will make this a little easier to see. Let's give it a shot again. Doesn't want to. There we go. So we'll make these a different color. So our polar microtubules are used to actually create the separate poles of the cell. And what these do is they kind of push against each other and they elongate the cell in preparation for final division. Remember, this is all right now still occurring. This is all occurring within our cytoplasm. And then we're eventually going to have what are called kinetochores. Kinetochore microtubules attach to the kinetochore of chromosomes. During cellular division, the cell membrane is going to break down. So there'll be no membrane surrounding the chromosomes that have condensed in the cell. And we'll talk about that when we get into the stages of mitosis. But these kinetochore microtubules attach directly to the chromosomes themselves. The chromosomes have been replicated. That's why they're in this X formation. And in that replication, the two sister chromatids, that's what these are called, right? These are copies of each other. The two sister chromatids are attached in the middle, somewhere in the center, by a set of proteins called the kinetochore. And at the kinetochore microtubules will attach. So here I have a microtubule attached on this kinetochore, and this microtubule is attached to this kinetochore, and these two chromosomes are going to be pulled apart so that this cell over here, we'll call this cell number 1 and this cell number 2, so that cell number 1 is going to receive These 2 chromosomes in cell number 2 will receive these copies, so that we have the correct number of chromosomes of each pair of chromosomes into the resulting daughter cells that are going to form following mitosis. So we have, of course, the centrosome. The centrosome is where these microtubules are coming from, and they are referred to as an MTOC or MTOC, which stands for Microtubule Organizing Center. So that's what they do. The single centrosome will duplicate at the beginning of the M phase of the cell cycle. So as soon as we go from G2 into the mitotic phase of the cell cycle, those centrosomes are going to replicate very rapidly. Another important distinction here is that animal cells have centrioles, but plant cells do not. Plant cells do not have centrosomes. They use their cell wall and other mechanisms for cellular division. And then, of course, to review, our 3 types of microtubules are the astral, the polar, and the kinetochore. So let's move into the different phases of mitosis. Here, what we're looking at here in this picture is this is interphase. And during interphase, this is just kind of a phase the cell will be in G2. This is a G2 phase. And when the cell is in G2, the chromosomes are kind of decondensed, and when they enter into G2, and you can see that the chromosomes are kind of spread out throughout the nucleus, right? Just as we get to the end of G2 to move into the M phase, those chromosomes are going to condense up. And that's actually what's happening here. You can see that these chromosomes are starting to condense, but notice they're still spread out in their territories. They are not necessarily in that kind of X formation that we are used to seeing. So prior to mitosis, the chromosomes, they do replicate, and that's what's happened here. We have multiple copies, that's why there's so many, and they'll begin condensing or kind of getting much smaller in preparation for division. Once mitosis, we've entered into the M phase and our centrosomes have replicated, we now move into mitosis. And in mitosis, we have 5 phases. We have prophase, prometaphase, metaphase, anaphase, and telophase. Directly following mitosis and the end of the M phase of the cell cycle, we have cytokinesis, which is division of the cytoplasm and, of course, the cell membrane, and in plants, cell walls. So let's look at our phases of mitosis. Here we have prophase. During prophase, our chromosomes have replicated. They've become much more condensed and tightened up. You can see here, right? We're starting to see actual chromosomal-like structures, kind of these little worm-like structures in here. So we're seeing that we've gone from 46 to 92 because our 23 pairs have replicated. Our nuclear membrane is beginning to break up. You can see our centrosome here, and all of these microtubules are starting to come out. And these microtubules are going to be used to go to pole. So we have both astral microtubules, kinetochore microtubules, and the pole microtubules. We have the chromatids continuing to condense, and eventually they will become super compacted structures. In prometaphase, we have the final breakdown of our nuclear membrane. We now have no nuclear membrane left. This is what this red stuff is here. These are all pieces of nuclear membrane that have been broken apart so that the chromosomes are now open to the cytoplasm. We have our 2 centrioles here, and the creation of, looks like, our kinetochore microtubules here. And astral microtubules will be coming out this way as these centrosomes are moving towards the opposite poles of the cell. Spindle fibers, so the mitotic spindle begins to interact with our sister chromatids. You can see now our chromosomes are fully condensed and replicated. So now we have very distinct chromosome-like structures, much easier to see. The more condensed they are, right, the more dense, the lighter the blue here. That's why we see the difference between the purple and the blue. The 2 kinetochores on each pair of the chromosomes are now attached. In prometaphase, our kinetochore microtubules are attached to the kinetochores of the chromosomes so that we can move into metaphase and line everything up. So now let's take a look at metaphase. Here we have interphase. You can see the differences here, just kind of give you an idea. Interphase. Here we have prophase. And here's prometaphase. So we can see the structural differences in each of these. Here we can actually see the opposite poles. Here we just have some, looks like pole microtubules. Our centrosomes are getting ready to replicate. That's why it's so bright here. Look, we have the two centrosomes here have replicated, and here they've just moved to opposite poles of the cell. In prophase, our cell membrane is still somewhat intact, but when we move out here to prometaphase, our cell membrane is no longer intact. So in metaphase, we now take those sister chromatids that have been replicated and are attached to each other, as well as attached to their kinetochores, and they line up. And this lining up is in the center of the cell. It's still a single cell. We just can see now our poles, our microtubules are moving to opposite poles, and our kinetochore microtubules have elongated. They're getting longer because these guys are pulling apart. And what's going to happen is they line up the chromosomes along what's referred to as a metaphase plate. Now, the metaphase plate is not an actual physical structure. It's an imaginary structure, and it's just kind of this dotted line that occurs halfway through the cell where the chromosomes are going to line up. And if I were to draw it here, here's my two kinetochores, right? And I have my, so these guys are kind of like this, right? So I have my two chromosomes there. And my microtubules, these guys on this side are going to attach there. These guys are going to attach here. And they're going to pull each of these chromosomes apart from each other. But the kinetochores line up along this plate so that all of my chromosomes are lined up this way, and they're sideways like this, so I can pull these two chromosomes apart. And when the alignment is complete and the cell is all completely aligned along this imaginary line or metaphased plate, the cell will then move from metaphase into what's referred to as anaphase. So here in anaphase, the connection between the sister chromatids gets broken. So that little complex, those kinetochores that would normally connect these two chromosomes together get pulled apart and separated. Now it's a single chromatid, and each sister chromatid, each copy of the chromatid gets pulled by the kinetochore microtubules to opposite poles of the cell. In order to do this, kinetochore microtubules, instead of being pulled like a rope, they actually shorten. And what they do is they constantly shorten from this range. So they'll break down but keep reattaching to that kinetochore on the sister chromatid to keep pulling it in this direction towards the opposite pole of the cell. The two poles themselves, these centrosomes, will move further and further apart. Right? So they're moving in opposite directions. They'll move further and further apart, and as they move further apart and our kinetochore microtubules begin to shorten, we start to see separation. So now we can see along that metaphase plate how these sister chromatids are separating. Eventually, we move into telophase, and in telophase, the chromosomes are at their respective poles. So we can see now that our chromosomes are completely separated from each other. I have two completely separate cells. My poles are visible, and remember, this is in the nucleus of the cell. My nuclear membrane is going to start reforming fairly soon. I have kinetochore and I have astral and polar microtubules here that are also helping to pull these two cells apart and start separating them. I now start seeing the reformation of the nucleus. You can see this red region right here. I have pieces of the nuclear membrane are going to start reforming, and I'm recreating the nuclear membrane. Each new nucleus will contain the 22 pairs of homologous chromosomes as well as its one pair of sex chromosomes. So here we have metaphase lining up on that metaphase plate. I like this little drawing here. It's a little easier to see. Then we have anaphase. In anaphase, we're actually pulling apart the chromosomes. And then in telophase, and they're referring to cytokinesis here as well, we're now reforming the cell itself. We're starting to create our new nuclear membrane. You can see that these guys have been separated, so I have copies of each of these chromosomes in opposite poles of the cell, and we're going to start moving into cytokinesis. Now, cytokinesis is observed when we start seeing what's called a cleavage furrow, like this. which you can start to see a little bit in this nuclear membrane here. So this is kind of a really a quick, neat video. It just kind of shows, this is a time-lapse video of mitosis in a cell under the microscope. So it's actual mitosis occurring. So we're in prophase, moving into pro-metaphase. The cell is kind of making sure that those chromosomes are copied and that they're going to be sorted out. The chromosomes are beginning to line up towards the metaphase plate. Replication, now that they're being pulled apart, we're in anaphase. You can see here we're starting to see that cleavage furrow, and the cell gets broken apart. Here's just another view of the same thing. Which was pretty cool. What's amazing is once they line up on the plate, how quickly anaphase occurs. So there's your metaphase plate. Anaphase, telophase, cytokinesis. And so now these two cells, look at how the chromosomes decondensed at the end there. And so in cytokinesis, cytokinesis happens at the very end of M phase in the cell cycle. That's the M phase there. Our two nuclei are completely separated into their two opposite ends. In animal cells, the cell membrane will form what's called a cleavage furrow. And in this cleavage furrow, it's kind of like taking a piece of string and wrapping it around the cell and then pulling it really tight. And that would constrict like a drawstring. You can think of it that way, that it's kind of restricting and pulling apart those two cell membranes. This is done with microtubule proteins. And then in plant cells, instead of a cleavage furrow, we get what's called a cell plate. So here we're at the end of telophase in this particular cell here. And the cell begins rebuilding the cell wall first. So we get a bunch of cell wall occurring down the middle, and this is referred to as a cell plate. And the cell plate will be the initial division between the two daughter cells. Eventually, the cell membrane that lines the inside of, like you can see right here, that's cell membrane. That cell membrane will then eventually line the inside of each of these so that the cell membrane is now intact. So some things to think about in terms of mitosis. These are some questions that you should review and use for study questions towards your exam. Why is mitosis important for multicellular organisms? What is interphase? And what part of the cell cycle are cells in interphase? Where in the cell cycle would a cell be in interphase? During mitosis, that's easy. That's the M phase. But what about interphase? Where there's actually more than one stage of the cell cycle there. What are the different stages of mitosis and what's happening in each? What is the difference between mitosis and cytokinesis? What are primary differences of cell division between plant and animal cells? There's more than one. What are poles in mitosis? What do we use the term pole for, and where are they located? What are kinetochores? What are they made of? And what are the three different types of microtubules that are part of the mitotic process? And how do chromosomes separate? What's the actual separation mechanism? What's happening between kinetochores and kinetochore microtubules? How are they shortening? How are they being pulled apart? And what is a metaphase plate? What's happening at the metaphase plate? Why is the metaphase plate important in mitotic division? It is very important. That metaphase stage is important for a reason. I want you to think about that. So that's our lecture for mitosis. Our next lecture will be on meiosis, and I will see you guys then.